In Chapter 23, we start the bulk of our diversity unit. We start by talking about prokaryotic cells and our prokaryotic domains, the archaea and the bacteria. And then we move into talking about our eukaryotic domain and the protists, fungi, and eventually plants and animals that come out of the protist group. In Chapter 23, I kind of divide it into two parts. One that is bacteria and archaea, our prokaryotic cells, and then part 2 will be our protists and our fungi. And then we will have whole chapters coming up later on plants, invertebrates, and vertebrate organisms. So for this set of lecture videos, we're just covering microorganisms, specifically prokaryotic microorganisms. So microorganism is just a broad term used to describe an organism that is so small you have to use a microscope to see it. There are no one type of cell that is a microorganism. Many of our protists are considered microorganisms. Some of our fungi are considered microorganisms. And it really just means that they're just super tiny. But most of the organisms that we consider microorganisms are going to be single-celled organisms. And this falls into the category of the prokaryotes, archaea, and bacteria, and then our single-celled eukaryotes, the protists. So we're going to start by talking about the prokaryotic domains, the archaea and the bacteria, and then we'll move into the eukaryotic domain after that. In Chapter 17, we talked extensively about the mechanisms of horizontal gene transfer. Horizontal gene transfer is going to be important for prokaryotic species because it's going to allow for genetic variation and therefore evolution to happen very rapidly in our prokaryotes. Prokaryotes are asexually reproducing organisms, which means they're not exchanging genetic information when they reproduce. This is in contrast to eukaryotes, some of our larger organisms like plants and animals, and some fungi that are actually exchanging and mixing genomes during sexual reproduction. So horizontal gene transfer is going to be an important mechanism for increasing the genetic variation within these major domains. But eukaryotes can also perform horizontal gene transfer. So when a eukaryote feeds or engulfs a smaller organism, brings an organism in by phagocytosis, they are also bringing in genetic information and could potentially take up some of the genetic information of that organism. And we saw this in Chapter 22 when we talked about endocytosis. Endocytosis is going to come back, and endocytosis is really important in our discussion about bacteria because two of our major organelles were came from the bacterial line. And so we can also have horizontal gene transfer when microbial organisms, small organisms, microorganisms, invade other cells. So we talked about viruses, how viruses invade cells. Bacteria and fungi can also invade cells and infect a host organism. And when that happens, there could be some genetic transfer as well. So horizontal gene transfer is a major evolutionary mechanism for prokaryotes, and it's important because it's going to increase that genetic diversity. When we're talking about prokaryotes, we're not going to see a lot of changes in their structural diversity. We are going to see some variation. We're going to see some variation in shape and some of the physical characteristics, but most of the differences in our prokaryotes are going to be metabolic, meaning the type of respiration they do, or whether or not they do photosynthesis, whether or not they can utilize oxygen, whether or not they can utilize specific resources or withstand specific conditions in their environment. So much of that diversity is actually going to come from horizontal gene transfer. So horizontal gene transfer is really important in early evolution of these organisms who are otherwise reproducing asexually, and depending on the only other source of genetic variation, which is mutation, which may or may not be helpful and may or may not occur very frequently. So our two prokaryotic groups fall into two domains. Remember that domain is our highest classification. In the protists, we're also going to talk about supergroups, but then after that, we have our kingdom, phylum, order, and so on down our phylogenetic classification. So you may want to review that because we are going to start talking about some of these different levels, some of these different groups. For prokaryotes, we are going to talk about archaea and bacteria, and we are not really going to go much lower than the domain level. We're going to talk about some specific examples, but otherwise we're generally going to leave the discussion of those specific phylogeny for a microbiology class. So prokaryotes is just a broad term that describes a cell type. From Biology 101, that cell type is a cell that does not have organelles, so it doesn't have a nucleus that is surrounded by a plasma membrane, although it does have DNA. And prokaryotes can do nearly all, sometimes more, of the metabolic functions than a eukaryote can do. So don't mistake a simple cell for one that can't do very many things or is not very highly evolved, because they have changed and evolved. Most of their evolution is just metabolic instead of the wide variety of structures and functions that we see in our multicellular organisms. So these cells, these prokaryotic cells, are the smallest cells that we know of. Thinking back to the cell theory from Biology 101, the cell theory tells us that the smallest living entity that we can have is a single cell. So since cells are relatively small, our smallest organisms are bacteria and archaea. But they are the most abundant. They account for half of the Earth's biomass. So even though they are incredibly tiny, there are tons and tons and tons of them. Bacteria and archaea inhabit pretty much every single environment on Earth, even the super extreme ones. So you might think it's too extreme for you. It is not too extreme for bacteria or archaea. Archaea is a monophyletic group. Bacteria is a monophyletic group. What this means is that all of the archaea and all of the organisms with the defining characteristics of an archaea come from a single common ancestor. Same thing with bacteria. All of the organisms that are considered bacteria with the specific bacterial characteristics come from a single common ancestor. And the branch with the archaea split off, and we had two prokaryotic branches. In our next set of lecture videos for part 2 of chapter 23, we'll talk about the eukaryotic group that split off from the archaeal group. But the archaea itself and the bacteria itself are considered monophyletic. As I mentioned, archaea and bacteria live in essentially every conceivable habitat that you can think of. This goes for all of the mundane ones like door handles, toilet seats, your gut, to super extreme ones like being encased in ice and around deep sea vents on the edges of volcanoes and sulfur or methane vents. So all kinds of places, because those organisms, while they are not significantly different structurally, they are significantly different metabolically, and they have adapted to all of these crazy environments and all of our normal environments by genes that allow them to utilize a huge variety of different resources. Archaea and bacteria are usually single-celled, but there are a couple of exceptions. Very commonly, both of these will be found in colonies or filaments. These are groups of single cells. Colonies are individual organisms that kind of are stuck together. We often see them as a film or a clump. And then many single-celled organisms also organize into filaments, which are long chains, like we see here on this Anabaena. Anabaena is one of the organisms that we look at in lab, and you'll see that it is a long, thin chain. There are a couple of different cell types, but they are still considered single-celled organisms. There are just some specific cell types that happen in the filament. They are the same type of organism, but they take a slightly modified form when they are in their filaments. And there are a few weird examples of bacteria that are truly multicellular organisms where they have specialized cell types, and those multicellular structures depend on one another. They depend on those cell types. I am not going to cover any examples here. If you are interested in it, you are welcome to Google it and look it up, but there are very few of them, and so again, we're going to save that for if and when you all take microbiology. So let's talk a little bit about Archaea. Archaea are more closely related to eukaryotes than they are to bacteria. And if you recall from chapter 22, we talked about the endosymbiosis theory that gives us the theory for the origin of some of the organelles in our eukaryotic cells. Now, it is hypothesized that the ancestral cell to the eukaryote was an archaea, and that archaea engulfed a smaller bacterial cell, what is the precursor to a mitochondria. And that means that the ancestral cell for the eukaryote was actually an archaea, and then that line of archaeal cells evolved into the eukaryotic cells that we know today. The reason for that hypothesis, that it was an archaeal cell as opposed to a bacterial cell, has to do with some molecular information that we have about the Archaea group. The histone proteins, which are the proteins that DNA is wrapped around in eukaryotic cells, are very similar to proteins in Archaea that also wrap up chromosomes. So we have to compact our chromosomes into really tight spools inside our cells in order to get all of that information in, and the chromosomes are wrapped around these histone proteins. Bacteria don't have histone proteins. Archaea do, and their molecular structure is relatively similar to the eukaryotic histone proteins, which suggests that an ancestral cell had that type of protein. Ribosomal proteins are also more similar between Archaea and eukaryotes than either one are to bacteria. Bacteria have ribosomes. Ribosomes are the structure in the cell that build our proteins, and ribosomes themselves are made up of two parts. They are made up of ribosomal RNA molecules and ribosomal proteins. And so when we look at the protein amino acid sequences for the ribosomal proteins between the two, they are much more similar in Archaea and eukaryotes than to bacteria. Again, suggesting that the ancestor to the eukaryotic cell was an Archaea, and then as evolutionary time wore on, Archaea cells diverged a little bit, eukaryotic cells diverged a little bit, but they still maintain many similarities. The same goes for RNA polymerase. RNA polymerase is the molecule that builds the messenger RNA when we are expressing our genes. So it reads the DNA template, copies it into messenger RNA. Again, similar RNA polymerase structure and protein sequences, DNA sequences related to the RNA polymerase in Archaea and eukaryotes. There's not nearly as much known about Archaea as bacteria. Bacteria have been much more extensively studied, mostly because bacteria are found in more normal locations. They're easier to collect and find. Archaea tend to live in crazy places and have just not really been studied quite as much. Many bacteria cause disease, whereas many Archaea do not. Most of them don't even bother us. So they just haven't been studied. As of now, there are around 20, a little over 20 phyla in the Archaea groups. If you're interested in these, again, you can go ahead and Google them for now or take microbiology, but even in microbiology, they don't go over a ton of detail about Archaea just because so little is known. Archaea tend to be extremophiles. Now, there are some bacteria that are extremophiles, but most of the archaea are extremophiles. In other words, bacteria tend to live in what we would consider more normal environments, less extreme environments, and archaea tend to live in more extreme environments. That's not to say that some of them don't live in normal conditions or what we would consider normal or more habitable conditions, but many of them exist in sort of these extreme areas. There are some bacteria that live in extreme places as well, so neither one is limited, but we do see a lot of archaea in some very specific environments. So areas that have, or water that has very high salt content can be home to some archaea. These archaea are called halophiles. And up on the top here, this is an image of a lake with basically a salty, solid structure that comes out of it. One of the famous places for this is a lake in Northern California called Mono Lake, and the structures there are called tufa, and tufa is basically really, really salty rocks, and the water around it is super salty, super basic, and just kind of a crazy environment where not a lot grows. Some archaea live in extremely acidic environments. Some live in environments with very high methane levels. And then we also have some organisms that are called thermophiles or hypothermophiles. These are organisms that live in really extreme temperatures. So these can be around thermal pools. This one down here on the bottom is a thermal pool at Yellowstone called Grand Prismatic. This here is called a black chimney vent. This is another extreme location that some organisms live in. And again, not all of them are exclusive to archaea or bacteria. It's just that archaea tend to live in some of those places. Another super salty lake that you might have heard of is the Great Salt Lake in Utah and also the Dead Sea in the Middle East. So just some kind of Although archaea and bacteria are both prokaryotic organisms, and although they are single-celled and have some similar characteristics, so for example, they all are single-celled, they have a plasma membrane surrounded by a wall, many of them have flagella, there are some very specific characteristics that distinguish the archaea groups from other prokaryotes. So the main thing are the lipid membranes and the type of cell wall that are found in archaea. Now, both of these, the cell membrane and the cell wall, help or have structures that allow them to live in these more extreme environments. We'll talk about the bacterial cell wall in another video. The bacterial cell wall and the bacterial cell are going to be kind of our standard that we're going to compare our archaea against. So we're going to kind of think of the bacteria as normal, and that's really just because it's more common. We know a little bit more about it. But archaea have very specific plasma membranes that are formed with ether bonds. So down on the bottom, we've got the phospholipid from bacteria and archaea. They are similar structures in the, bacteria and eukarya. They have a phosphate group at one end linked with a glycerol molecule and 3 fatty acid chains. Now the bacteria and the archaea membranes are a little bit different, but for our purposes, we're going to talk about the difference between bacteria and archaea. In the archaea phospholipid, we have these additional bonds that you can see here compared to the same location down on the bacteria. These are structures that basically are little side chains that make the phospholipid just a little bit different. And then the bond that we have in the top of our molecule is also a little bit different. So there's an ether bond, which you can see it's a carbon to oxygen, as opposed to an ester bond, which is a carbon to two oxygens. And the ether bond in the archaeal cells makes the archaea membrane more resistant to heat and extreme conditions. It's just that the chemistry is a little bit different, changes the stability of the molecule, allows it to interact a little bit differently. Then we've got these chains. Again, we've got these side chains. They're just a little bit different. They are made up of what is called isoprene, which is a modification to the hydrocarbon tail. So instead of our fatty acid tails, we have these isoprene chains. So just a little bit different, allows them to function in different types of environments. And then the cell wall of the archaea cell are a little bit different than the cell wall of our bacteria. Bacteria cell walls are made up of something called peptidoglycan. We'll talk about that when we get to the bacteria section. The archaeal cell walls are made up of proteins, more so than they are carbohydrates. So peptidoglycan is a carbohydrate, and the cell walls of the archaea are really more made up of protein, a higher protein content. So we don't talk too much about archaea, even in any of our other chapters, including our ecology unit where we talk about global warming. But one of the important sort of functions of archaea is that some of them produce methane. So they break down carbohydrates in certain environments, one of which is in the guts of some of our ungulates. That's why we have our little cow friend there. And when they do, they produce methane. So one of the things that cows happen to do quite a bit is pass gas, because they are constantly eating foods that are really hard to break down. And so bacteria and archaea in their guts actually help them break down methane, but they also do it in other environments as well. So archaea groups, many groups within the archaea phylogeny are going to release methane. Some of these live in swampy wetlands, so that's kind of why wetlands and swamps are really, really smelly. We've got a lot of methane. The bottom of lakes, so these are anoxic environments, environments that don't have any oxygen. And then also deep sea habitats where there might not be a lot of oxygen down there. And then, of course, in the digestive system of some of our animals. The reason that this is important is that methane is a more potent greenhouse gas than carbon dioxide. So we talked about carbon dioxide mostly in our ecology unit, or if you're in a spring semester, it's coming. We're going to talk about carbon dioxide and greenhouse gases. But methane is much more potent, meaning that it has a higher warming potential than carbon dioxide. And so what that means is that for the same amount of methane as a unit of carbon dioxide, you get warmer temperatures. So this is really important if we are trying to limit our greenhouse gases. One of the things that we can do is limit the amount of cattle that we are grazing, and our other herbivores, sheep, goats, etc. We can limit some of those because some of them are producing a lot of methane. So we mentioned that the archaea are more closely related to the eukaryotes than the bacteria are. We've seen this slide before. This is the horizontal gene transfer slide that shows the transfer of some genes across between bacteria to bacteria, bacteria to archaea, and then bacteria to the eukaryotes. This also shows some of the important bacteria that we're going to talk about. We're going to talk about the cyanobacteria and the proteobacteria in our bacteria lecture, but it also shows some of the archaea groups. One of them that it shows is called the Asgard archaea. The Asgard archaea are considered to be the most, share a recent common ancestor, recent being very long time ago, around at least 1.8 billion years ago. So it's been a long time, but the most closely related group is called the Asgard Archaea. All of the organisms in here are named after Norse gods, which is kind of entertaining if you want to look these up. But they are related to eukaryotes. And we talked about the histones, the ribosomal proteins, and the RNA polymerase. There are other proteins in the Asgard Archaea that are even more closely related to the eukaryote than the general ribosomal RNA, et cetera, that we talked about at the beginning of this lecture. So there are a couple of protein types, those involved in vesicle trafficking, meaning those that are moving vesicles around a cell. Vesicles are like little pockets of membrane that move cell components or cell products around a cell. And then also those that are important in the regulation of actin, which is one of the cytoskeleton filaments. So the cytoskeleton is made up of 3 different types of filaments. One of them, the smallest one, is called actin. And there's been some information or research done on the dynamics of this structure, and it appears that the proteins that the Asgard Archaea use for a cytoskeleton are similar to the proteins that the eukaryotes use. So there's some crazy, crazy science going on here, a lot of molecular information that tells us that these archaea are more closely related to eukarya than to any others. I'm sorry.